In this demonstration, we'll be illustrating how SynBioHub can be utilized to exchange genetic design information between various software tools. In particular, we'll be using SBOLDesigner, Benchling, and ICE. And we'll be using the SynBioHub instance that's hosted at Boston University for the Living Computing project. Let's first take a look at the collections that we have on the Living Computing SynBioHub instance. In this instance, we have a collection of the cello parts that were used in the science paper that was recently published. These parts were recently converted into the Synthetic Biology Open Language, or SBOL. In addition, we also have the two ICE registries and the link to the Benchling parts for the CIDR lab. One thing that's important to note is that the ICE registries and the Benchling parts are private. That means that a user must have special permissions to be allowed to see this data. While on the other hand, the cello parts are public parts, which are visible to anyone. Let's first take a look at the cello parts. So in the cello parts, we see that there's 166 entries, and let's look at one particular part, the A1 AMTR. So in this description of the part, we have some meta information. We also have a diagram for the part. If you hover over, you can see that it's composed of a ribosome entry site, a coding sequence, and a terminator. There's also a reference for describing where the part came from. You can download the SBOL for the part, or the GenBank for the part. You can also find where the part has been used, and find identical parts, and send the parts to Benchling and ICE, which I'll be demonstrating shortly. But first, I'd like to build a larger design out of this part. And to do that, I'm going to use the SBOLDesigner tool. So in the SBOLDesigner tool, I'm going to instantiate a promoter, followed by this part of interest. To add this part, I'm going to select to fetch it from the registry. And now it's bringing up all the matching parts, including the A1 AMTR part, which I'm going to select, which adds it to our canvas. Next, I'm going to select the promoter for this design. I go into the library, and I once again find all the promoters available. I select this promoter to add to our design. Now, at this point, if you'd like to see more information about the parts, you can hover over them. For example, for this A1 AMTR, I can select it, and I can browse inside, and you can see that matches the way it shows in the SynBioHub registry. I can then add some metadata describing the part, that it's a genetic inverter, setting its ID, name, and description fields, and you can see that the DNA sequence has been derived. At this point, I'd like to send the part to SynBioHub so that I can share it with our other tools. So to do that, I select the SynBioHub programmingbiology.org instance, select OK, provide my username and password, and upload. And now the upload has been successful. So let's go back to the SynBioHub now and take a look at the part that I've just uploaded. To do that, we click on Submissions, and it brings up the genetic converter part that I've added to the repository. So here we can see the part that I've uploaded, and let's select the particular part. And you can see that it's a hierarchical design composed of a promoter and this A1 AMTR part, and you can see below what's inside that part. Now at this point, I'd like to do sequence level editing, and the sequence level editing tool that we like to use is called Benchling. So I click Send to Benchling, and now it's sending the genetic converter part over to the Benchling repository. And this view actually shows all the parts that I have in my SynBioHub transferred folder at Benchling. So I can select this part, and you can see in this case the part has been flattened because Benchling doesn't support hierarchy, and so we give it a flat image of the design. So now I select Edit, and it's going to take me over to Benchling where I can do my sequence level editing of this design. Here's the sequence level view in Benchling, and I can do whatever editing and other sequence level work that I wish to do. Once I've concluded that, I can come back here, and I've now completed my sequence level design, and let's assume that I've built it in the laboratory, and I'd like to store information about where this construct is available in my laboratory. To do that, I'm going to use another tool called ICE. To use that tool, I click on Send to ICE, and it's going to send this information about this genetic converter design to the ICE registry. So now it's added this to the list of the parts that I have in the Living Computing ICE registry. You can see it is shown here. I select that. You have some additional information. And if you select this link, it'll take you to the ICE registry. Now within the ICE registry, I can view the sequence with a couple different viewing mechanisms. View it on a plasmid view. I can also store information about the sample, so I can add the sample. Say it's in a 96 well plate. And say sample. Submit that information. So now let's go back to SynBioHub. And at this point, I've finished and I want to now add some more information about the experiments I've performed on this part. And to do that, I'm going to use the annotation features within SynBioHub. But first, I need to copy the part to SynBioHub. So I can select Copy to SynBioHub. And now it appears in my private submissions within SynBioHub. I can go into this ICE collection, find my genetic converter design. And at this point, I want to add some additional information about it. Perhaps I have some transfer function that I'd like to add. So I can select an image, add that to my design. I can go add some comment, plot of my transfer function, add that to my design. And I can add additional notes, source, or for example, I can add a PubMed ID, which will go look that up on PubMed and find the citation for it. At this point, I'm happy with my design. It's complete. So I'm going to go back to my submissions, and I'm going to make this public to share with everybody. So the design is now available publicly, which means that it can be searched upon. So if you put into the search criteria information about the part, it'll show up in search queries like so. And that concludes this demonstration.